I'm going to talk to you today about two processes that occur in the cell called transcription and translation. Okay. These are both continuous processes that occur in the cell. Transcription is the process of changing DNA into RNA, and translation is the process of creating proteins from RNA blueprints. Okay. There are a few differences between DNA and RNA. The first one is that DNA is double-stranded, while RNA is only single-stranded. A second one is that DNA has one less oxygen than RNA, and RNA has one more than in the ribose sugars. And a third one is that while the DNA uses the four nitrogenous bases of cytosine, guanine, adenine, and thymine, RNA does not use thymine as a complementary base pair, and instead of thymine it has uracil, and uracil replaces T, so it's a complementary base pair of A, and C is still complementary with G in RNA. Okay. The three parts of the RNA process are initiation, elongation, and termination. And in the cell, DNA is stored in the nucleus, and all RNA except for mRNA is stored in the cytoplasm. And RNA is also in the nucleus with DNA. So, in transcription, DNA strands go from the 5' to the 3' direction. And when proteins are needed for the cell, they need to be transcribed and translated for DNA. The RNA polymerase starts the sequence of transcribing DNA and RNA, and it has something called a primer. Which is the start sequence, and it tells when to start, so this could be the primer. And during the transcription of DNA into RNA, the base pairs are switched along with uracil and thymine. So, for instance, if the DNA pair was what could be A C T, it would translate in mRNA, which M stands for messenger. It would translate to A would become U, G would become C, and T would become A. So that is transcription for you. At the end of transcription, the mRNA leaves the nucleus and enters the cytoplasm, where there are many ribosomes, and it gets ready for the transcription process, where RNA will be translated into proteins. RNA blueprints will be translated into proteins. Thank you. I'm going to talk to you guys about translation. Sorry about my handwriting, it sucks. Okay. Translation. Translation is the process where RNA is changed into proteins. So, in order for RNA, translation basically starts when the mRNA leaves the nucleus, where the DNA also is, and it goes into the cytoplasm. It binds on to these little different ribosomes, and remember that ribosomes are where the proteins are made, and it wants to translate into proteins. But proteins are made up of many amino acids, so it's a lot easier if you use the simple, if you translate the mRNA, the RNA into amino acids. So, what happens is that, the mRNA is translated into individual amino acids, and it's carried and brought by the translator where it happens, and which is called tRNA. And at tRNA, these I'm so brain dead. At tRNA, the RNA codes are put into groups of 3, and these are called codons. And every codon can be translated using the codon chart with different amino acids. So let's say, well, I know AUG, which is the starting codon, which I'll talk about in a second. And what else is there? Say ACU. So then using this codon chart here, I can tell that because it's ACU, which I just lost. Okay, it's THR would be the amino acid, and AUG is MET. I'm so tired. I'm sorry. MET, sorry, I was right. Okay, MET. And so, the tRNA reads the mRNA in these triplets, and it initiates something called start and stop codons. And so this is the protein chain, and it's always going to start with the codon AUG or MET, and you will always know that the protein chain is ending when you see UAG, UAA, or UGA. And at the A site, these codons are added. So then there's something called an A site, which stands for amino site, and that's where the protein chain is elongated. And it's elongated, so it'll take something that's in its mRNA code, in its mRNA state, and it will change it to its tRNA, and then from the tRNA, the protein chain reaches its length, and there's like lots of different codes of letters. So this is one code, this is another, and it keeps going, and the protein is made when it is all in its amino acid translation. Okay, and so as an overview, I'm just gonna like review. Oh, and there's one thing I know. Oh, and the rRNA creates the chain of proteins. Okay, so as an overview, I'm just gonna think of some example from the top of my head and show you how just briefly demonstrate this. So let's say the DNA code complementary base is AGT. Okay, AGT. The mRNA would change this mRNA into UAC, and then the tRNA would further change this into AGU. So then it's pretty similar to this, just with the letter here. And the last amino acid for UCA is SCR. So this is, and you use the coding chart. So that is how you, so that is basically how you transcript and how you translate. And I'm really sorry, and change DNA into RNA and translate RNA into proteins. Thank you for watching.